The purpose of this video is to cover the topics in Chapter 14 related to biotechnology and genomics. In the course of this lecture, we will talk about the goals, examples, and tools of biotechnology. And some of the examples that we'll be tools that we'll be talking about are DNA cloning, polymerase chain reaction, and gel electrophoresis. We'll then talk about how those tools are used in biotechnology to make copies of DNA, how they've been used in particular cases to make certain products of biotechnology, and then at the end we'll talk about, briefly introduce the idea of gene therapy and cover some of the basics of genomics and proteomics. So the first thing that we're going to do is talk about how we can clone DNA. When you clone something, you're making exact copies. And when we're talking about genes, we're making exact copies of the pieces of DNA. And that's what it is to clone a gene. Very simple, just making a copy of something. And when we're talking about the use of it in biotechnology, we'll talk about two different tools that can be used to clone genes. The first is called recombinant DNA technology. And we'll talk about recombinant DNA technology, which is shown in this picture here. The second tool that we'll talk about is polymerase chain reaction, or PCR. So to begin, we'll take a look at recombinant DNA technology and how it can be used to make exact copies of genes that a scientist might be interested in. In the case here, we're seeing human insulin. So human insulin, the idea here was to produce human insulin outside of a human, package it, and then it can be used to treat people who are diabetic. In order to do that, scientists made copies of the gene that codes for human insulin and then utilized bacteria as little molecular factories to make that insulin protein for them. In order to do that, they had to make copies of the insulin gene and get it into the DNA of the bacteria so that the bacteria would have the code to make that protein. So a couple things have to happen first. First, you have to identify the gene in the human genome that codes for the human insulin protein. Okay, so we're not going to go into the details of how that works, but once you identify where the gene starts and stops, you would be able to use some tools to take that insulin gene out of the human DNA and put it into the DNA of the bacterial cell. Now we've talked about the differences between eukaryotes and prokaryotes, and here we see a eukaryotic chromosome that's linear, and we see the prokaryotic chromosome that's circular. In addition to the normal prokaryotic chromosome, circular chromosome, bacteria often have little accessory rings of DNA called plasmids. So here's a plasmid right here. These plasmids are much easier to work with than the genome of the bacterial cell, so they're easily removed from the bacteria, they can be manipulated, and then they can be replaced back into a bacterial cell very easily. And we'll actually be doing this in lab upcoming in a few weeks. So the idea here is take the human insulin gene, take the plasmid, and splice these two pieces together. Much like we saw splicing with introns and exons, you're just piecing two separate pieces of DNA together. In order to do that, we need to talk about some tools called restriction enzymes. So one way to splice two pieces of DNA together is to first cut the DNA and then link the two pieces together. And to do that, we have some tools called restriction enzymes and another tool called DNA ligase. Restriction enzymes, as enzymes, they're proteins, and what they do is bind to DNA and cut the DNA. Now there's lots and lots of different restriction enzymes, and each will cut the DNA at a very specific place called a restriction site. And for example here, this restriction enzyme will bind to the sequence GAA-TTC and cut that in such a way that it produces this staggered cut. Okay, most restriction enzyme sites are palindromes, so you'll see GAA-TTC on the top, and if you read the bottom strand backwards, it reads the same, GAA-TTC. That's a very common restriction enzyme. But what you see here is it cuts the DNA in a way that it leaves these staggered ends and cut it instead of cutting it right in the middle. What you're able to do is then cut the human insulin gene with a particular restriction enzyme, cut the plasmid DNA from the bacterial cell with the same restriction enzyme. In doing so, it creates these compatible sticky ends. For example, this right here would be the insulin gene, and this right here would be the plasmid DNA. Okay, and if you cut them with the same restriction enzymes, what you see is the sticky end with the insulin gene will match up with the sticky end of the plasmid DNA on both sides. And so it's like a puzzle piece that fits together. However, another enzyme, DNA ligase, is needed to seal these two pieces together. This DNA ligase is the same enzyme we saw in DNA replication that sealed together the Okazaki fragments on the lagging strand of DNA replication. So this DNA ligase has the potential to bind these strands, create these covalent bonds between these sticky ends, and create a single uniform piece of DNA. Now, this plasmid DNA is mostly bacterial DNA, but now it has this human gene inserted into it. As such, we call this recombinant DNA. It has been recombined, some human DNA, some bacterial DNA, together into one piece of DNA. So it's been recombined, and so we call it recombinant DNA. Any two sources of DNA that have been put together by DNA ligase, for example, can be called recombinant DNA. Going back to our schematic then, we now have the human insulin gene and the plasmid DNA. Both were cut with restriction enzymes. That's what we see in step 1. Step 2, then, if you use DNA ligase, you can seal these two pieces of DNA together and you have recombinant DNA. This recombinant DNA, what we can do is we can induce a bacterial cell to take up that recombinant DNA. You can kind of trick it into taking up DNA from the environment and incorporating it inside the cell. Now, this unique thing about this plasmid DNA is that there's other genes on it as well. And so the bacterial cell recognizes this plasmid DNA and it'll transcribe and translate the genes that are encoded for on that plasmid. And so as we've introduced the insulin gene now, the bacterial cell will start making copies of that gene or cloning that gene. Every time a bacterial cell divides, remember it has to make a copy of its DNA. It makes a copy of its genomic DNA, and it also makes a copy of the plasmid DNA. So here what we see is the cell has divided twice, and every time you see that little purple piece of DNA, this gene has been cloned because we've made exact copies of that gene in new cells. Not only will these cells be replicated, these genes will be replicated along with the host DNA, but it'll also be transcribed and translated to produce insulin. Using biochemical methods, then you can extract and purify that insulin, and once it's purified, it's the human insulin protein, and it can be packaged up after it's been purified, packaged up, and then marketed for those who have diabetes and need to have insulin injections. So this method of recombinant DNA technology allows us to recombine pieces of DNA from different organisms and then induce bacteria to make that protein. What we'll be doing in lab is taking a piece of plasmid DNA that has mostly bacterial genes, but also has the green fluorescent protein gene, so the gene that's going to code for the green fluorescent protein. We're going to take plasmid DNA, we're going to take some E. coli, which is a type of bacteria, and we're going to induce that E. coli to take up the plasmid DNA and then induce it to express the green fluorescent protein. And what we'll see is bacteria then that glow green. We talked about the idea that a transgenic organism has foreign DNA in it, so we'll be making transgenic bacteria because it has a foreign piece of DNA from a jellyfish. These also represent transgenic organisms because they have a gene from humans incorporated into the genome of the bacterial cell. I encourage you to now go to the course website with the textbook companion site, and there are two videos from the textbook website that I encourage you to watch. One is called The Steps of DNA Cloning, which will walk you through this video to walk you through these steps of recombinant DNA technology. The second video is called Restriction Endonucleases, and it'll talk about restriction enzymes. So I encourage you now to take a minute to watch those videos and then come back to the lecture video here. So we just talked about how we can use recombinant DNA technology to make copies of a gene or to clone a gene, using bacteria as little molecular machines to do that for us. Now what we're going to talk about is another technique called polymerase chain reaction. And polymerase chain reaction is a method where we can, again, make copies of a piece of DNA, so copies of a gene or clone a gene. Instead of using little bacterial cells to do that, we can do it in a machine called a PCR machine. The premise of polymerase chain reaction is to take the DNA, particularly a gene that you're interested in, and go through several rounds of replication. Okay, so here, the semi-conservative method, you see one blue old strand is maintained and a new red strand is made. From there, if you split those again and make new green strands, split them again and make new yellow strands, what you see is after a few cycles you have lots and lots of copies of a particular gene that you're interested in. The way that this differs from normal DNA replication is that we can target a specific gene that we're interested in, so we only replicate that gene and we don't replicate the rest of the DNA. There's a website by the Dolan Learning Center that sponsors different kinds of animations for molecular biology. And I think they have a really good animation to help us understand polymerase chain reaction. You can also access this video on YouTube.
Polymerase chain reaction, PCR, enables researchers to produce millions of copies of a specific DNA sequence in 1 to 2 hours. This automated process bypasses the need to use bacteria for amplifying DNA. A PCR reaction starts with a denaturing step. Samples are heated to 94 to 96 degrees Celsius for 30 seconds or more to denature or separate into single strands the target DNA. Next, the temperature is lowered to 50 to 65 degrees Celsius for 30 seconds or more. This allows the left and right primers to anneal to their complementary sequences. The primers are designed to bracket the DNA region to be amplified. The temperature is raised to 72 degrees Celsius for 30 seconds or more. This allows the Taq polymerase to bind at each primed site and extend or synthesize a new DNA strand. The temperature is raised to 94 to 96 degrees Celsius, denaturing the target DNA as in cycle 1. As before, the temperature cools to 50 to 65 degrees Celsius, allowing more primers to anneal. The temperature is again raised to 72 degrees Celsius. Taq polymerase binds to each primed site and synthesizes a new DNA strand. In subsequent cycles, the process of denaturing, annealing, and extending are repeated to make additional DNA copies. After 3 cycles, the target sequence defined by the primers begins to accumulate. After 30 cycles, as many as 1 billion copies of the target sequence are produced from a single starting molecule.
A couple of things to highlight. First of all, is that these primers that are used are very specific to the gene that you're interested in. So you would be able to design primers. There's machines that can make little pieces of DNA like this. Design DNA primers to start at the beginning of a gene here and stop at the end of a gene. And so you can flank your region of interest using these primers, and so that when replication occurs, you're really only replicating this region of interest. Just like we saw with DNA replication, there's little primers that need to start the process so that DNA polymerase can then work. So you flank your region of interest with these primers, then you use a version of Taq polymerase that allows these ends to be extended through replication. And so using several rounds of this heating up to 95 degrees, which will denature the strands of DNA, breaks the hydrogen bonds that hold the base pairs together, so that denatures. Then you lower it to about 60 degrees Celsius or so, 65 degrees Celsius. And what this does is allow your, lower it to 55 degrees Celsius, allows your primers to anneal, anywhere from 55 to 65. So that allows those hydrogen bonds to form between the primers, which are in excess, and the DNA. Then you raise it to 72 degrees Celsius, which allows the DNA polymerase to do its work of extending the ends of the DNA for the top strand, for the bottom strand here. So what we see with PCR is we have some DNA that needs to be replicated, and let's say this is our gene of interest. What we then do is can design primers that'll bind. So first of all, let's say this is the 5' end of the DNA and the 3'. That means on the complementary strand, it's anti-parallel. DNA replication occurs in the 5' to 3' direction. And so here, a primer would bind here, and this would be the 5' end of the primer and the 3' end of the primer. And so replication would occur in this direction. You would also have a primer that would bind down here. This would be the 5' end of the primer and the 3' end of the primer, and replication would occur in this direction. And so after the first round, you've made 2 copies of your gene, right? You started with 1 copy and you've made 2 copies. Now normally this first strand continues to extend as long as it has template to work with and as long as the reaction still goes. I'm going to use the bottom strand as an example for the next round. So we have the full 3' to 5', right, showing here. We also have this top strand that starts somewhere here at the beginning of that primer and extends all the way through the end of the template. Now the next round, what we'll see is that primers will bind here and here, right? So this is the 3' end of that one, and so this would be the 5' end of the new strand, and it would proceed in this direction, and this would be the 5' end of this strand, and it would proceed in this direction, making a copy. And so then we'd have 2 more copies on the bottom, and the same would occur on the top. Now what you'll notice is this one here on the bottom will only extend until it runs out of template. And so this bottom strand that's made, this new bottom strand here, 5' to 3', is a very short strand, and it represents only the gene of interest. So what we see here is now we have only the size of the gene without excess on either end, right, of extra DNA, only the gene of interest. Now as you go through many rounds of PCR, what you'll find is that more and more copies of this, what we call the target DNA, will be created, and eventually a second strand of that would be created as well. And you'll get lots of copies of target DNA and only a few copies of DNA with extra pieces on the end, or extended DNA. And after only about 20 rounds of PCR, 20 to 30 rounds of PCR, which is very typical, you get overwhelming number of copies of that target DNA, millions of copies. And so that gene has been cloned. When this process was first discovered, there's a scientist by the name of Kary Mullis, and kind of an interesting story actually. He used a lot of drugs, and he claims to have determined the process of PCR, kind of envisioned the process of PCR while he was high on LSD. And so who knows to this day what would have happened if Kary Mullis hadn't determined this process. But at the time anyway, what they did was they used water baths. They would put the DNA in a water bath that was 95 degrees. They would allow it to denature. They would then move it to a water bath that was around 60 degrees, allow those primers to anneal, and then they move it to a water bath to allow for the DNA polymerase to work. And so it was really time consuming. Then what they discovered was a unique enzyme called Taq polymerase. And the thing that's unique about this is it comes from bacterial cells that live in hot springs. Okay, so they're used to living at elevated temperatures. And what we know about enzymes is each enzyme has a very particular temperature where it works best. Well, because this organism lived in these hot springs, this particular enzyme, this Taq polymerase, was very heat stable. Right? It had to work at high temperatures, so that enzyme was very stable at high temperatures. And so what that allowed, normally what would happen is every time you heated the DNA back up to 95 degrees, it would destroy the nature of the Taq polymerase. But when they started using this heat-stable Taq polymerase, then they could heat it up to 95 degrees and go through a cycle without that enzyme ever being destroyed. So they didn't have to keep replacing the enzyme that was used after every cycle. Suddenly, it became much more affordable to do this reaction because you weren't going through excess amounts of this enzyme. So Taq polymerase is commonly used in PCR reactions today. We also have machines now that will change the temperature for us, so you don't have to sit there and move it between water baths. It has a hot plate that then raises and lowers the temperature for a certain amount of time to facilitate cycling through these 3 different temperatures that help a single replication round to occur. So in the end, we end up with lots and lots of copies of our gene of interest. So here again we see the PCR. We start with a single strand or single copy of the gene of interest, and through PCR, 1 round of PCR, we make 2 copies. Each of those 2 copies makes 2 copies. And so exponentially, after many rounds of PCR, we end up with millions of copies of that particular gene of interest. So what do we do once we make copies of that gene? Well, we have to be able to look at that gene or then utilize that gene in different ways. One of the ways to analyze DNA after polymerase chain reaction, after restriction enzyme digest, things like that, is to run the DNA out on an agarose gel, which separates DNA by size, and then you can analyze that piece of DNA. So DNA gel electrophoresis is another common tool in biotechnology. Again, it separates DNA based on size. And so we use this compound called agarose, and you can it's kind of like gelatin, so you can make a gelatin mold of this agarose, and it has little holes in it, and the holes allow the DNA to move through the gel. But larger pieces of DNA can move more slowly than smaller pieces of DNA. So DNA is negatively charged. This is based on the sugar phosphate backbone. That phosphate in the backbone has a negative charge, so overall the DNA is negatively charged. So what you can do is apply an electrical current to this gel so that the DNA is pulled towards the positive pole. And the smaller fragments will move much more quickly than the larger fragments. And so you can end up separating DNA based on size. And you can see lots of different bands here. This gel kind of represents DNA from different sources, a mother, a child, and I guess in this case we're looking at paternity because we have two males, probably trying to determine who the father is. Because the child would have got some of its DNA from its mother, some from its father, we can then analyze the child's DNA to see which of the two males it matches. The idea here is that the DNA has been cut first with restriction enzymes. We all have similar DNA, but we have differences between us in our DNA that make us unique. Because of those differences in the DNA sequence, restriction enzymes will cut each of our DNA samples differently. So here, if we cut with the same restriction enzyme, these would represent mother's DNA fragments, where that enzyme has cut it into pieces. We can cut child's DNA, and then we can take the two fathers, potential fathers, cut their DNA. So what we see here is that certain bands from the child match mother's DNA, which makes sense. Half the DNA comes from mother, half from father. The other bands, then, what we find is that some bands in the child match male 1, but they aren't consistent with male 2. So what we can say is that male 1 is likely the father in this case. So by using gel electrophoresis, we are able to analyze these fragments of DNA from restriction enzyme digest, but we also can take the DNA from a PCR reaction and run it on a gel and separate those fragments by size as well, to see if we've actually made copies of the gene that we're interested in. A couple of other resources I'd like to share with you. First of all, again, on the course website for the textbook companion site, there's two animations that might be of interest. One is another example, animation to talk about polymerase chain reaction. The second is a video called DNA fingerprinting, and so that'll talk about some uses of this particular technique for using restriction enzymes to fingerprint people, like we saw with the example before of the paternity case. The third is a very good animation from the Dolan Learning Center, just like we saw with the PCR. So you can either Google Dolan Learning Center and gel electrophoresis, or you can go to the website that's listed here, and it'll walk you through the steps of gel electrophoresis. So I really encourage you to watch those videos as you need to further solidify your understanding of the steps of DNA fingerprinting and use of gel electrophoresis to separate DNA based on size. In class in a few weeks, we'll have the chance to set up gels and separate DNA based on size and do an analysis much like a fingerprinting test. So we'll get the opportunity to do that in lab. When we meet next for class, we'll also be doing a recombinant DNA technology practice set. And so that will be your in-class credit for your study guide. So most likely Monday we will do that first thing as we discuss any questions you have about these techniques. For the next few minutes, I'd like to give you some examples of ways in which biotechnology has been used. So I just talked about how insulin can be produced in bacteria and then purified and packaged up to be sold on the market. Other examples in bacteria, we're talking about genetically modified organisms, bacteria specifically, that have been modified. One example shown here is for bioremediation of an oil spill. Bacteria has been modified to be able to eat oil, basically. It has the enzymes necessary to break down oil into smaller components, and so they can use this modified bacteria, release it into the environment to help clean up oil spills, and so that's called bioremediation. Bacteria don't naturally eat oil, so they have to be modified to do so. Another example in bacteria is strawberries are really susceptible to frost, and so what they've done is to produce bacteria that have an enzyme that allow it to prevent ice crystals from forming, and so they grow this bacteria in the presence of the strawberries, and then it prevents this frost from forming on the strawberries. So again, these bacteria don't normally resist frost, but in this case it has enzymes that help prevent water crystals from forming into ice crystals. There's several examples of genetically modified plants. One of the classic examples is Flavor Saver tomatoes. These used to be on the market in the 90s. The idea is that tomatoes are very susceptible to rot from fungi. And so what they did was to modify these plants to be able to produce an enzyme that would prevent polyglucuronase. And so this prevented the fungi from growing very well. And so they called Flavor Saver because it prevented the rot once it got to market. And so you can see the control tomatoes here that don't have the Flavor Saver gene, and then the Flavor Saver tomatoes, and they last longer on the shelf. And so this, in 1987, there's a company called Calgene that got the patent for this, and then they got FDA approval in 1994, but it was removed from the market due to decreased yield. So even though it was a good thing post-harvest to have this gene, it caused a decrease in yield, so they weren't getting as much from the tomato plants as they expected. The patent expired in 2007, and these haven't really been on the market for a long time. Here's an example of Arabidopsis. Arabidopsis is a model system that's used to study plants, and these Arabidopsis plants have been engineered or genetically modified to contain an extra gene to allow for a sodium-potassium ATPase. And so what this allows is for these plants to tolerate high levels of salt. So down here on the bottom we see Arabidopsis that have been treated with different concentrations of salt, and they're growing just fine. The normal Arabidopsis that doesn't have this special gene, when you treat it with salt, higher concentrations of salt, it really inhibits plant growth. So think of Wisconsin roadsides in the winter, dump all that salt, plants starting to grow in the spring with excess amounts of salt in the soil. Plants don't do well, and there's a lot of different agricultural soils that happen to be high in salt. So the idea here is to engineer plants that could withstand these high levels of salt and still grow fine. Another classic example of a genetically modified organism in plants is what we call golden rice. It's basically rice that has a gene to increase the levels of beta-carotene. Beta-carotene is really important for eye development, and in communities where a lot of especially poor Asian communities, where rice is basically the main substance in the diet, there's 250,000 children that go blind every year, and 1 to 2 million with weakened immune systems that die from vitamin A deficiency. So vitamin A deficiency is a really serious condition. The idea behind golden rice was to engineer these plants to produce high levels of beta-carotene. Beta-carotene is the precursor for vitamin A, and thereby increase the amount of vitamin A in the diet of individuals who really rely on rice. This work took 8 years to complete, and it was really important for biochemists trying to understand metabolic pathways about how vitamin A was produced. One of the main problems with golden rice at this time is that there wasn't enough enough beta-carotene to counteract vitamin A deficiency. And so the problem was that people had to eat a lot of rice in order for it to make a difference, and obviously they didn't have the money to eat that much rice. Right now they're working on an improved version of golden rice that has even higher levels of vitamin A. So right now this isn't really on the market anymore, but in the future it could help with problems like vitamin A deficiency. Other examples include most of the corn and soybean on the market in the United States have been modified to be resistant to Roundup Ready, which is an herbicide, and then for the corn to be resistant to the corn earworm. So it has a natural insecticide that's produced in it. So a lot of the food on the market today has been genetically engineered. Just to give you an example of how a plant might become genetically engineered, we've seen how bacteria can take up plasmids from the environment, but plants are a little different. Here what we have is a mature plant that is susceptible to an herbicide. So herbicides kill weeds. In this case, it can kill any plant. The idea is to make this plant resistant to this particular herbicide. So let's say you're growing corn in the field and a lot of weeds are growing around it. If your corn was resistant to an herbicide, you can spray the herbicide on the field, kill all the weeds around it, and then your corn wouldn't be harmed. And so what you do is take some tissue from this plant and you can culture that tissue in a media. Just like you would grow bacteria in a media, you can do that for tissue as well. And eventually you'd get single cells. And then what you can do is treat these cells with a bacteria that has the gene you're interested in, which would be for herbicide resistance. Bacteria, there's certain types of bacteria called agrobacterium that are able to pass their genetic information into the plants. And so what you do is you treat these with this bacteria, they introduce this gene of interest, this herbicide resistance, into the plants, into these single cells. Then you can treat these cells with that herbicide, and anything that has incorporated this gene would be resistant. And so you kill some of the cells that aren't resistant, that didn't incorporate the gene, and you'd be left with the cells that have incorporated the gene. You can take these cells and culture them into plant embryos. The embryos would grow into a plant and eventually into a new plant that now is tolerant of being the herbicide being sprayed on it. So it's resistant to the herbicide. So this is one of the ways in which you can generate transgenic plants. There's other mechanisms as well, but this is very commonly used for both soybeans and corn and other types of plants that are currently genetically modified. In animals, one of the examples of transgenic animals is that we have a gene for human growth hormone. And human growth hormone would stimulate growth in organisms, but it also, in some organisms, would stimulate the production of milk. And so what you do is you take this gene and you introduce it into an egg of, in this case, we're looking at goats. And so you would take the egg, the gamete, the egg of this goat, and inject the gene for human growth hormone into it. And then that gene could be incorporated into the genome of the goat, and you would implant this egg into a surrogate, okay, a host goat, who would then allow that, by planting it into a surrogate, that allow that egg to develop into a fetus, and then this donor mother would give birth to this transgenic goat that now has the human growth hormone. This growth hormone then would induce milk production in the goat, and you'd have milk that now is producing human growth hormone that would be present in the milk. So the positive side of this is that you get increased milk production. The downside potentially is that the milk may possess the human growth hormone in it, which then as we drink this milk, it could affect our development as well. So we've seen examples of genetically modified bacteria, plants, and animals. And we should also extend the animal examples to humans. And so in humans, we would call this gene therapy. The idea is not to create transgenic humans or genetically modified humans per se, but to use it for gene therapy. The idea is that there's a gene that's not functioning properly in a human, that it can be replaced by a functioning gene and thereby alleviate certain disorders. There's two possible ways to do this. First of all, direct injection, by directly injecting genes in a certain region. The other is to remove a cell, like a stem cell, modify that stem cell, and replace that stem cell. Any cell that then divides from that stem cell would have the modified gene in it. So you'll see here some examples, first of all, in the brain, Huntington's disease, Alzheimer's disease, Parkinson's disease, genes that are involved in those processes that may have mutations or aren't functioning properly. We could then replace it with a gene that is functional by injection. With skin, gene transfer by modified blood cells, so removing blood cells, replacing them, and thereby perhaps treat certain instances of skin cancer. In the lungs, perhaps the possibility of introducing new genes by aerosol spray to treat cystic fibrosis or hereditary emphysema. in liver by having modified implants to treat for certain liver diseases, blood through bone marrow transplants to treat sickle cell anemia, with the endothelium, which is the blood vessel lining, to implant cells that would have modifications that would treat hemophilia and other endothelial disorders. Muscle by direct injection to treat muscular dystrophy, and then bone marrow to modify the stem cells in bone marrow to treat things like sickle cell disease and other examples. So these are just potential applications of gene therapy, and in your near future, I guarantee there will start to become options for diseases like this as scientists begin to uncover how to successfully treat disorders through gene therapy. The last thing this chapter covers are some various fields within biotechnology, including genomics, proteomics, bioinformatics, functional genomics, and transcriptomics. So I'm going to give you some examples of what these are to understand how different groups of scientists use biotechnology. One of the main fields is genomics, and there's different types of genomics. There's structural genomics, there's functional genomics, there's comparative genomics. But structural genomics really focuses on what is a gene. And we've been talking about this in class already, that a gene is normally preceded by a promoter, has a series of introns and exons, and then it ends with a terminator. In the genome then, in a single chromosome, there would be lots of genes interspersed by what do we call it, intergenic sequences. Here we see gene A, gene B, gene C, all spaced out along a single chromosome. We also see what we call intergenic sequences. So these are just regions of DNA in between that apparently do not code for any genes. As we can see in the central dogma then, we have transcription, where pre-mRNAs are made. And then we have mRNA processing here, where introns are removed, and 5' caps added, 3' poly A's tails added, and you would have 3 different genes here. So the goal of genomics is to understand what the genome of different organisms looks like. So for example, you can examine all the chromosomes in humans, get a sense of where all the genes are, and trying to understand what each of those genes are doing. These intergenic sequences we used to think were junk DNA, but now we know there's a lot of information going on in those intergenic sequences. Some of it is repetitive DNA elements, some are transposons, some are unknown sequences. So some of it is junk DNA that we don't know what it's doing. Some of it may actually have a function, and we're continuing, as in the field of genomics, continuing to understand what's in those intergenic sequences. In 2001, the human genome was published. Its complete sequence, but not only that, but using computer tools to analyze that sequence and determine where those genes are, so that would be structural genomics. The field of utilizing different computer tools to look and answer questions about genomes and different features through biotechnology is called bioinformatics. So incorporating computer technology with what we know about genes. And through bioinformatics, we have certain websites. Here, the human genome is freely available on the internet through a website which allows you to browse the human genome, find out where different genes are and what those genes are doing. And so putting all of the information from the human genome in a website that's accessible for all researchers around the world. And so you can go through using this website and browse the human genome. So that would be an example of bioinformatics, using different computer tools to make these tools available to researchers. We also have what's called functional genomics, and the idea with functional genomics is you know where the genes are, but what are their functions? So how do all the genes work? Where are they expressed in the cell? What do the proteins that are produced from those genes do? So now we have the tools to try to examine all of the genes in the genome and figure out what they're doing, and that is the idea of functional genomics. And it uses a technology called microarray technology, which I'm not really going to go into right now, but if you're interested, I'd be happy to talk to you about that. It's the idea of looking at genes, figuring out from that the DNA. Microarray technology allows us to see what RNA is produced. If the RNA is being produced, we know those genes are turned on and likely have a function at a particular given time in an organism's development. So microarray technology allows us to look at the RNA that's present in an organism at a given time. With the tools of biotechnology, we've been able to sequence the genomes of many, many different organisms, look at structural genomics, look at functional genomics. And now we have the tools to do what we call comparative genomics. Comparative genomics allows us to use the tools of bioinformatics, these different published sequences, what we know about structural and functional genomics, to then compare how these are working in relation to other organisms. So here we have a segment of the genomic sequence where we would have different genes here, spaced by intergenic sequences. And what we're able to do then is to compare where these genes are positioned, which organisms have these genes in common, and help us to try to discern what the function of those genes might be. And if they function differently in different species, how does that lead to the overall differences that we see between organisms? So we've taken a look at genomics and bioinformatics, and now we can take a look at transcriptomics. So transcriptomics utilizes these tools that I mentioned called microarrays. Microarrays are a way to compare RNA that's produced at a given time point. And so you can look at time points under one treatment versus a control or no treatment, and you can see the differences in the RNA. And what that would tell us is when genes are turned on, what genes are turned on in response to a particular treatment, or what genes are turned off in response to a treatment. So what that allows us to do is to see what genes are working in different pathways and what those genes are actually doing. I'm not going to go into the details of exactly how a microarray works, which is what this slide is essentially showing, just to understand that it's a way for us to look at levels of RNA which would indicate when transcription has occurred and genes are on, or when genes get turned off. Considering the central dogma, then, we can look at genomics to look at DNA, we can look at transcriptomics to look at RNA, and then we can also use proteomics to look at which proteins are expressed at a given time in a cell. Remember, proteins do the business of the cell, so if you want to know what a cell is doing, one of the best ways is to look at what proteins are produced and at what levels to understand what might be going on in the cell. And so that's the field of proteomics. You can look at which proteins are expressed, what is the structure of the proteins, how do proteins interact with one another, and then using the tools of bioinformatics, compare these to get a sense of what is going on in the cell at any given time. These bioinformatics tools, then, as you can see, are not only accessible for genomics, the structure and sequence of DNA, but also what genes might be expressed at a given time and what their proteins are doing. And all of this, it gets incorporated into websites to help researchers who are researching a particular protein get a sense of when that gene's turned on or off and what it might be doing, and freely shared among researchers in the community. Lots of different organisms have their own bioinformatics sites that allow people to continually update with new advances in the fields. So with this, we have talked about examples of biotechnology, tools of biotechnology, and the goals, really, by understanding genomics and transcriptomics and proteomics, the goals of biotechnology. The goals of biotechnology are to understand what genes are doing and use our knowledge about those genes to find ways to improve different biological processes for bacteria, for plants, for animals, and for humans. And so the whole goal really is to improve different aspects of life in the medical field, in the agricultural field, and in other areas of our life. So we've seen now examples of recombinant DNA technology, polymerase chain reaction, and gel electrophoresis. We talked about some of the products of biotechnology, though there's no way have we exhausted that list. And now we've seen some of these different major fields of biotechnology. When we meet next, we'll have a chance to ask any questions about this material, give us some time to practice with some of the concrete examples of biotechnology, and then we'll be moving on to the next chapter. So thank you.